Well, thanks for that introduction. As you heard, I'm going to be exploring the evolving role of medical geneticists in the era of gene therapy, and calling out an urgency to prepare. And this came from an article that was just published in Genetics in Medicine earlier this year. I've put the reference up here. And recognize my co-authors. Always like to put up my disclosures for talks. So there they are: research funding and some consulting. And I'll just take a second to say that the NBSTRN is actually near and dear to my heart. I was the chair long ago of the advisory committee for it, and have continued to collaborate over the years in various projects. And as I hope you'll agree by the time this talk is finished, that you'll see that newborn screening through whole exome sequencing is a viable option. It's coming. I'm certain that NBSTRN will continue to play a role as in newborn screening and helping with that evolving technology. So with that, I'm gonna start with some statistics. For those of you who are geneticists watching this, you will know these statistics. 3% of infants are born with a genetic problem. 0.5% of babies have an inborn error of metabolism. The thing that most surprises non-geneticists when I talk about this topic is that 40% of infant and childhood mortality is caused by a genetic disorder. And depending on how you define it, up to half of childhood hospitalizations and a quarter of adult hospitalizations are related to a disease that has at least a significant genetic component. So it's a huge burden on the healthcare system. If those are surprising statistics, these are disturbing statistics. We've got about 10,000 rare diseases now on the latest numbers that are floating around out there, but very few are currently identified by newborn screening. Right? We've got roughly 3 dozen or more diseases on the current iteration of the recommended uniform screening panel, the RUSP. Perhaps even more disturbing is that of those 10,000 rare diseases, less than 10% have a treatment, and in most of those, the treatment is not curative. It just takes care of the symptoms. It's symptomatic relief, and so we've got to get to a point where addressing the cause of those rare diseases, and that brings us smack into gene therapy. And so I'll be talking about gene therapy and its role in genetics in the future here, because, well, I've been doing this stuff now for about 40 years, and the gene therapy's been just around the corner for as long as I've been doing it. Let's take a step back and view the classic path of new drug development. There's an early stage in the laboratory where someone is throwing lots of compounds at some assay that makes them think that it will be relevant to a particular disease. This filters down to a handful of test compounds, and then one which eventually comes out to be go into clinical trial. And this process usually takes over 10 years. It is undirected, and it's inefficient, and it's expensive, and especially true for rare diseases where the population across which a drug is going to be used is relatively small. The good news is that from this step at the top of this funnel down to this step at the bottom of the funnel for rare disease is actually much faster, and it's directed rather than undirected, and that's because in rare diseases we have usually a much better handle on the pathophysiology, and so you can design the treatment to actually address the pathophysiology and bypass sort of the random screen in many cases. the end result is, if you look at the latest data from the CIDR website, the Center for Drug Evaluation and Research, what you can see is that for probably the last 10 years anyway, the orphan products that have been approved by the FDA have outnumbered, or at least are equal to, the number of drugs that have been approved for everything else in medicine. That's kind of a remarkable achievement, and I think that gene therapy is only going to move that forward faster, and that discrepancy is going to become even larger, just because there are now 10,000 diseases out there that need a gene therapy. Now, just to remind you why things have been moving so slowly in gene therapy, it really is is a result of both the need to identify and build drugs one at a time for one disease at a time, but also some problems that have arisen. Most of you are familiar with the unfortunate case of Jesse Gelsinger, who died in the middle of a gene therapy program for ornithine transcarbamylase deficiency. What you can see relative to newborn screening, which started in 1963, the first enzyme replacement therapy was in 1991. The first mRNA replacement therapy, which was for spinal muscular atrophy, was in 2016, and the first FDA-approved gene therapy for a retinal dystrophy was in 2017. So there really is a long timeline from the start of newborn screening to the present. But this really is, this really is going to change. Because I think we are turning a corner. If you look at the readily identifiable gene therapy trials on clinicaltrials.gov, as of the middle of 2022, when we were putting that article together, there were 334 potentially durable non-oncologic gene therapies in development, 40 of which were in phase 2 clinical trials. And the expectation is that a current passage of those through the approval stage, that by 2030, we're likely to have 30 gene therapies approved, addressing 50,000 patients. And so you can see here in a graphic how that plays out. And the question now becomes, how do we get there from here? How do we get to being able to scale gene therapies and make them accessible and not overwhelm the system? And that's what what I'm going to spend the rest of the time talking about here. The implementation of gene therapy in a way that allows us to not get derailed or from a purely from the standpoint of the profession of medical geneticists, to not have us become irrelevant to the process. So if you think about the process, the first thing you have to do to be able to treat something is to identify it. And I think the diagnostic odyssey over the last 5 to 10 years has really become much more publicized, and people are aware of the fact that most genetic disorders are only identified after a long delay. And that's because functional testing is really only available for a limited number of disorders. The things that we do as screening for genetic disease mostly address metabolic disease, and even then it doesn't pick up everything. And of course, we are now at an inflection point where whole genome sequencing is becoming cost effective, and it can, in theory, identify any single gene defect in patients who you're trying to diagnose. And in the context of newborn screening, anything else you want to add to that menu. And I contend that we are at the point where whole genome sequencing really just needs to be our primary diagnostic test. That's not to say there aren't other things that can or should be done, shouldn't be done beforehand, but ultimately every patient walking into a genetic office deserves an answer. And they deserve it sooner rather than later. And whole genome sequencing is the cheapest alternative to get us there. Not only that, but it is available to analyze in the future, and sets up the dynamic of what I call age-appropriate screening rather than newborn screening. Because you can go back and analyze this at any time and identify diseases that are appropriate to know about, say, for example, when you're 20 years old, as opposed to when you're 2 days old. So let's look at medical geneticists and gene therapy. There are a whole series of issues that need to be addressed as we move forward on this, and we'll talk about things, and depending on where you begin on this, well, I guess it's not a wheel, it's a hexagon, right? Starting with screening, and then the regulation that approves gene therapies, the collaborations that are necessary to implement them, scaling to meet the needs of 260 million people rather than the 20 that are in a clinical trial, and then issues around how these are going to be delivered, whether that's a centralized version or a decentralized version, and how we engage people so that this is done in an equitable way, as opposed to just only those individuals who can afford to pay out of pocket or have good insurance that covers it. So no gene therapy, no therapy can get to the market without getting through the regulatory agencies here, the FDA and the EMA in Europe called out. And we're at an inflection point with them as well. Unique trial designs really need to be available for rare disease therapy, because double-blind placebo-controlled trials are often difficult and sometimes unethical in the context of a genetic disease. And those N+1 trials that we do when we think we know something works and we manage to treat a patient somehow don't translate into clinical trials very well, because as soon as you add the second patient, that's not just 2 N-of-1 trials, it becomes a full clinical trial, and the amount of work that goes into that becomes exponentially higher than just by adding a second patient. Once you do manage to get a gene therapy approved, what does that mean? What is gene therapy? Well, if you look at most gene therapies that are coming out in clinical trials right now, some of them address other organ systems, but a lot of them are focused on the CNS or the liver. So, is that simply an infusion that you get in an infusion chair for an hour, you get your vector, and you walk away, or you get your intraventricular infusion through a lumbar puncture? Is it a one-time procedure? Might it have to be done again? What else is necessary for that? So is it a single chair in an infusion center, or is it an entire system? Who will pay for it and who will do it? And so let's take a look at that. If you look at models of gene therapy delivery, they're not so different from most of what else we're doing. I mean, you can think about a very centralized system where there is a central hub and spokes around it, and the spokes represent centers that refer to a hub, maybe can do some of the components of a gene therapy protocol, but not all of them. And so they're integrated, and then those hubs all coordinate as to where they how they assign patients. It's a little bit like what happens with liver transplant, for example, right now. You can set up a liver transplant program if you have all of the resources. But you also need to be able to provide all of the therapies around those diseases. When we get a transplant here in Pittsburgh for a urea cycle defect, we have to be able to care for the patient beforehand, during the transplant, after the transplant, and do long-term follow-up. Alternatively, you can have centers that focus on diagnostics, treatment, follow-up, and some of them will have some of those, one or more of those components. Some of them will have all of them, and work through coordinating centers. This is something that's going to need to be worked out, and in a way that allows efficient delivery to the maximum amount of patients with the least amount of disrupt in their everyday life. Who's going to get gene therapy? Well, they have to be selected appropriately. And this is really one of the reasons why I think medical geneticists need to play a central role in gene therapy. And it's because to give gene therapy, you've got to have the right genotype. You have to be giving the right gene therapy for the right gene, the right and potentially even the right mutations at the right time. Otherwise, you're wasting money. You have to understand the phenotype of the disease, and after all, that's what we do, right? We're master phenotype individuals. That's what we do in our evaluation, and understand the spectrum of disease and when gene therapy is appropriate. We need to evaluate other therapeutic options and engage the other specialists we need to help us out. Maybe we need the cardiologist to help us treat the cardiac symptoms until the gene therapy takes hold. Maybe the cardiologist will want to be the person that gives those gene therapies, but probably not. And so that's going to be in collaboration with us. And coming back to equitable distribution of healthcare resources, these are going to be $1 million or $2 million drugs. How are we going to give those out? How are we going to get those to everybody in a way that doesn't break the system? I've got way more questions than I do answers, but I'll have some ideas about some of these for you as we go through the last few minutes of the talk. Education is going to be a critical component of gene therapy, and this is both teaching patients and their other caregivers about the therapy, our own genetic staff who are going to be helping us deliver these, the other clinicians that are part of our gene therapy programs, hospital administrators. Ultimately, you got to set these things up in a hospital, and they need to understand why they're important. Insurers have to be able to pay or be willing to pay for these, and the pharmacists have to deal with a different category of drug than they've had to in the past. I like to use the comparison to liver transplant, which of course is a gene therapy. You're giving a whole new liver, a whole new genome. Estimates in our institution is that a liver transplant over the course of the first year, immediately before the transplant and the year after the transplant, probably amounts to about $1 million in billing to the hospital or by the hospital, and that the hospital gets maybe $75K in operating margin out of that. The reason is all of that other money, 95% of the money that's coming into the system is used for infrastructure. Well, if a gene therapy is nothing more than an infusion chair, that's an hour in infusion chair is probably $1,000 or a couple $1,000 in our current reimbursement system. Is that all we're going to get for delivering a $2 million drug? I don't think so. I don't think it makes any sense. But how and if that's the case, how much do we need to be able to say, I've got to go to my liver transplant folks, and I'm saying, you're not going to be doing OTC liver transplants anymore. I'm going to take care of that with gene therapy. The hospital loses that income from the procedure and only gets a couple $1,000s from an infusion. Probably not tenable. So my proposal for this is that reimbursement for gene therapy should be 10% of the cost of the drug. Now, I'll let you niggle as to whether that should be 20% or 5%, but I think 10% is reasonable. Why? Well, as I said, we're the ones that have to figure out that this is actually the right drug to give. If it's the wrong drug, then insurers, hospitals, and drug manufacturers are wasting their money. They're getting the wrong drug to the wrong person at the wrong time to take everything in that first sub-bullet and negate it. The patient needs full phenotyping and appropriate pretreatment care. If you're going to be delivering, I'll go back to my OTC example. If you're going to be delivering gene therapy to OTC, you want to be sure that that patient is in optimal condition before you get to the therapy. Then you have to be familiar with delivering it. What are the infusion and treatment reactions? And monitor for the immediate post-treatment complications. We're seeing a lot of press now on TMA or atypical HUS following AAV gene therapy, but there also are patients that have died of liver failure because of a liver-based gene therapy. So you have to be prepared to deal with those complications. And then you don't want to use a $1 million drug if you don't think it's going to be effective in the long term. And so long-term follow-up is necessary for this to happen. And I contend that telling an insurer that I'll deliver this $1 million drug and make sure it's done right and that you're getting good value for your dollar for 10% of the cost of that drug. I've been getting some interesting comments back on this, and when I present this proposal to our own insurers and others across the U.S. when I've been speaking. Now, if we go back to looking at the who's doing this, I contend it should be geneticists for the reasons that we've just discussed. Are there enough of us? This is just from the ADMGG website as of yesterday. And it says that there are 1,860 board-certified MD clinical geneticists in the United States. Now, that doesn't say that all of those are practicing. Some of those are retired because this is just anybody who's ever been boarded. And the last time I tried to get a real number on that, it's probably around 1,200. Can we do all of that gene therapy? I think the answer is probably no, and we need to increase our workforce. How do we do that? Well, this comes down to our friends at the ABMGG and the ACMG. I am currently a board member of the ACMG, and there's a lot of concern about the workforce. And I think this comes back to changing the view of what medical genetics is. We have forever been viewed as diagnosticians. And in medical school, a lot of genetics courses are not taught by geneticists. What students see is Down syndrome and making a diagnosis by clinical features and maybe karyotype, but they don't see that our primary test is whole genome sequencing. I mean, how can it get any more exciting than that? And students need to see that. And now they're not seeing that medical geneticists are de facto developing into therapeutics specialists. We're already doing it with enzyme replacement therapy, but we have other therapies that are coming down the road. So as medical geneticists, we need to embrace and publicize our role in therapeutics, and that this is really a primary endeavor for medical geneticists. Now, broad-based diagnostics shouldn't be ignored. In fact, we should be promoting it as the rule and not the exception. Everybody gets a genome, not just the people who we can get insurance for or who we don't understand what's going on. Maybe there's some, you make a diagnosis without it, that's fine. But most cases, it's going to come down to being the exome or the genome as the fastest way to a diagnosis. Gene therapy should become a core competency in MedGen residency training. That's a heavy lift, and it will take some doing, but I think we need to do it. This is not just a handful of medical geneticists who are comfortable with it who are going to need to be the ones to do these therapy. We have to sell genetics to medical students. There's no way around this. We have to be able to get it, capture those students before they decide they want to be surgeons or cardiologists or GI docs. We have to be part of a strong national advocacy for gene therapy. And ultimately, if we're going to be presenting this to other specialists, care providers, families, advocacy groups, ourselves, we need educational materials that we can use in our MGG training, as well as to train others. So in summary, all I can say is gene therapy is here. It is actually here. We're doing it. And medical geneticists are uniquely positioned to be the providers of gene therapy. We should be conducting clinical trials. We can and should be coordinating implementation. We can be the leaders of the educational care team, and we should be integrating with multidisciplinary teams to deliver this effectively and safely. MedGen training has to embrace therapeutics as well as diagnostics, and that diagnostics has to be broad-based. And I can never resist tossing out some challenges to the FDA that they've got to be able to recognize novel trial designs for approval of drugs for rare diseases, because the current ones are just not. We can't do double-blind placebo-controlled trials for most of these conditions, and we cannot approve drugs for one disease at a time. That's another talk. So with that, I always like to thank my lab and my collaborators, and especially my co-authors on that original paper that I showed. And with that, I thank you. And as we go back to the live portion of this, I think we'll be moving to questions and answers. So thank you.